Hello everyone. Please confirm if I am audible to you. And are you able to see my PowerPoint? Are you able to see the PPT? Yes. Good evening everyone. Welcome to the Brahmastra series once again. Okay. So here we have our first question. Today is Unit 5B, where we will be discussing fertilization and cleavage divisions. Okay. So we have studied it already in the classes, and let's see the different questions which have come in CSIR net from these two topics. Okay. So this is something regarding mammalian fertilization. Please try answering it. Okay. So we have human sperms are allowed to fertilize ova having non-functional ovastasin, right? So as we know, this is a molecule which is released from the egg after fertilization is completed. The following possibilities may be of significance in the fusion of these gametes, that is the sperm and the egg, the male gamete and the female gamete. So what will be the possibility, which combination of statement represent the outcome of the above event? So you have to answer whether the statements are true or false, keeping in mind that the ovastatin is non-functional. Okay, ovastatin doesn't work properly. Okay, so what will happen now? Ovastatin, what is that one thing that it will do? It acts as a block to polyspermy. Okay, if you know that it acts as a block to polyspermy. Now we are talking about mammals, so that's why we should be keeping in mind that what is our egg membrane. So in mammals, the egg membrane is zona pellucida. Plasma membrane is there, but additional membrane is zona pellucida, which consists of the glycoproteins. One is ZP1, ZP2, and ZP3, right? Now, ZP3 is the major glycoprotein which is required for recognition with the sperm proteins, and ZP2 is required. I mean, ZP2 is also required in the later process, but at a later stage, the ZP2 is actually cleaved. Okay, it is cleaved by the ovastatin. This is what happens. So why it when what happens when it is cleaved? Ovastatin, when it is cleaving the ZP2, since I said it is required for the recognition, right? It will be needed for the recognition of the recognition of the sperm and the means the sperms binding to the egg membrane. Okay, they should be binding to it and penetrating the zona pellucida and getting inside the egg. But we don't want polyspermy. So what is polyspermy? When many sperms are fertilizing the egg. Okay, so to avoid that, the ovastatin cleaves of the ZP2 so that any more sperms are not able to fertilize the egg. That's the function of ovastatin. Now, when it is released, we know that after fertilization, there is a cortical granule synthesis. I mean, cortical granule exocytosis, where the granules of uh, the cortical vesicles, which are there in the cortex, those undergo exocytosis. Means their contents are released outside the cell, in between the zona pellucida and the plasma membrane. Okay. So from there, it will interact with the zona pellucida ZP2, and it will clip the ZP2. Okay. So if you see over here, yes, Rahul, that's right. That's the right answer. Female mice lacking ovastatin does not cleave ZP2 after fertilization. and mouse sperm bind equally well to ovostasin lacking 2-cell embryos as to normal eggs. Okay, so you can see here these are the layers which are shown, zona pellucida. Actually, this is what your zona pellucida looks like, the structure, where you have this blue color. These are the ZP3 molecules, and on top of them you have the N-acetyl glucosamine residues. Okay, to which the molecules come in, to which this sperm receptors will be binding. So this is the sperm receptor known as GALT. Right? This is a sperm head. So those will recognize as ZP3. And here ZP2 is also present, and cross-linking them, there is a ZP1 also. Okay, so this is the zona pellucida, how it is interacting. Then you have the egg, as you can see here, some sperm is coming and binding. So we have ZP1, ZP2, and ZP3. So here we have the glycan release glycosidase. These are all from the cortical granules. What is happening? So if the glycan release is there, just have a look what happens. There is a disulfide linkage somewhere here ZP2. So that part is actually cleaved. Okay, there is a breaking in that part of ZP2, and in ZP3 also some changes will be there. If you have a chemoattractant, so you can see with the chemoattractant the sperm is able to come to ZP1, so there no changes are happening. But if you see here, once this ovastatin is released, okay, so that ovastatin, because of which what is happening, you can see this part is completely clipped off. Okay, it has been taken off. So as a result, what happens, the sperm is not able to proceed any further. Got it? It will not be able to proceed any further. So these are the ways how fertilization can be stopped. Yes, Saujanya also right. So in chemoattraction, what happens? You dissipate the gradient. That means you change the gradient. We know that the gradient will be more towards the egg, right? But if you change the gradient to somewhere else, then also the sperms will move away. They will not be able to bind. Okay. And if glycans are there, they are directly binding to the sperm head, so they will not be able to fertilize the egg. So as you can see, this disulfide bridge here, it has been broken. Got it? So if there is non-functional ovastatin, then yes, ZP2 will not be clipped by the cortical granule protease. And also, if ZP2 is not clipped, then many sperms will be able to bind and enter the egg at the same time, and even in a small amount of time. So polyspermy will be occurring frequently. Okay, so C and A is the right answer. The sperms will not fertilize over. Why not? If ZP1, ZP2, and ZP3 are intact, there is no reason that sperms cannot fertilize. Sperms will fertilize, okay? Because you don't have the blocker. If blocker is not there, then definitely all the fertilization processes will take place. The sperms will bind and penetrate the zona pellucida, but will not be able to fuse with the ovum membrane, okay? CD9 protein of egg membrane microvilli will not be able to interact with sperm membrane proteins in the absence of ovastatin. So CD9 is, we know that it is required for the fusogenic protein. So it is present on the egg. Why it will not be able to interact with sperm? We are not doing anything to the egg. And in fact, ovastatin doesn't do any changes in the egg microvilli. So they will still be able to interact with the sperm membrane proteins even in the absence of ovastatin. Okay, got it? Let's move on to the next question. So before moving on, let me inform you about the Watson Junoon scholarship, as you might be all aware of. So we have extended this one. So 15th August is the last date to apply because of high demand. Okay. So what you're getting along with this high 1-year coaching for CSIR NET and GATE exam, or even you can have QET IIT JAM and GAT-B exam. 12 months of access to all global and national workshop. As you know, we have already conducted one workshop this month regarding big data biology, and we have one more upcoming in the end of this month. And these workshops, they are conducted by even global speakers from all around the world, and even from India also we have eminent speakers who join this workshop. So you will be getting access to all that along with the scholarship. Okay, our programming internship also you will get. So all this you are getting in a package with live project hands-on certification courses. You would know from our website, you can select of your choice 4 certification courses. You can select whichever you feel will be required in your resume. You can add them and you can access them. Direct access to our coaching experts on WhatsApp and one VIP pass to our CRISPR conference, which will be happening in 2023 this year, which is a mega event. It's called as CRISPR Congress 2023. Okay. So you have to apply now in case you want to join us. You can either call us at our toll free number or you can use this number. So if you're calling anytime outside office hours, then you can contact in a toll free number only. Okay. So this then next is number 2 question. Which one of the following statement is incorrect? Transient rise in calcium is necessary for egg activation in mammals. Sperm induces egg activation and does not involve calcium. In many organisms, eggs secrete diffusible molecule that attract and activate sperm, and capacitated mammalian sperm can penetrate the cumulus and bind the zona pellucida. So which one out of this is incorrect? You have to select the false statement. Okay, very good. So, most of you have answered correctly. Very good. Surinder, Deepshika, Rahul, Achal, Aishwarya, Anupam, Suraj. Yes. So, the answer is sperm induces egg activation and does not involve calcium. Because we all know that egg activation, the major signaling molecule is calcium. So, this is an easy part B question, but just wanted to show you that you do get a combination of questions also. Some people are saying that developmental biology, some tough questions will come, but it's not like that. If you prepare properly, there will be some easy questions as well, which are direct. Anyways, part B questions are always direct. Okay. So we do have a transient increase, means in a wave there will be an increase, not all at once. Okay. Yes, Anupam, Saujanya, Tejashwini, all of you are right. And definitely, yes, secrete the molecules which are called as chemoattractants that will activate sperm. And capacitation is a process whereby the sperm becomes matured to fertilize the egg. So they can penetrate the cumulus layer of cells and they can bind with the zona pellucida. Okay. So as we have already seen the signaling process, which happens after the sperm has fertilized egg. So we have the PIP2, which is cleaved into IP3 and DAG. DAG goes and activates the sodium proton pump. IP3 takes out calcium from the endoplasmic reticulum, and that in turn activates the cortical granule exocytosis and also leads to the egg activation indirectly. Okay. Coming to the next question. For successful fertilization in sea urchin, interaction between the surface of the egg and acrosomal proteins, specifically a 30.5 kilodalton protein called binding, is necessary. The following factors could affect this interaction and prevent fertilization. So, which out of this combination can prevent that interaction between the binding and the egg surface? Okay, we'll see. Anupam, Surinder, Rahul. So I'm getting different answers. Let's see whose is correct. Okay, Saujanya also says 3. Okay, most of you are saying 3 only. Let's see. So what is it saying? That between the surface, egg surface and binding, we need the interaction, right? For successful fertilization. So if you remove binding, isn't it? That can prevent fertilization, isn't it? If you remove binding, that can remove fertilization. If you remove binding receptors, that can also block fertilization, isn't it? But where is binding present? It is already mentioned over here. Yes, it is already mentioned. It is an acrosomal protein. Now, acrosomal protein means what? Where it will be present. So this is a nucleus. This is the acrosome. So where will acrosome be present? It will be present on the sperm, isn't it? So acrosome will be present on the sperm. So it's an acrosomal protein binding, which means binding is present on the sperm. So if binding is present on the sperm, binding receptor will be present on egg only, right? Correct. So that is one thing. Now whether it is present on the egg vitelline membrane or it is present in the egg jelly, that is our question. Okay, I'll come to that one. Another one is removal of egg jelly polysaccharide. So we know that egg jelly layer, it is required for chemoattraction. Correct. So if the sperm doesn't come to the egg itself, then how can it be interacting with the egg membranes, isn't it? So egg membrane, you have first the plasma membrane, then you have the vitelline envelope, and then you have the jelly layer, isn't it? So first it crosses the egg jelly where it undergoes acrosomal reaction, then it enters in the through the egg jelly and it will encounter the vitelline envelope, isn't it? So if you remove egg jelly polysaccharides also, you will not have the interaction. Okay, so yes, many of you are right. Aishwarya, Suraj, Saujanya, Surinder, everyone is right. It is option 3, A and B. Okay. And yes, as I mentioned, on the egg jelly, you don't have binding receptor because binding is a fusogenic protein. So they will be only interacting when it crosses the egg jelly. Fine. So they will be present on the egg vitelline envelope. Fine. And it's not like from a single cluster. If you remove from single cluster, then other binding receptors are already present, isn't it? So you have to remove all the binding receptors from the egg vitelline membrane. Got it? So we have already seen this diagram in our class. So as you can see, the sperm is first encountering the egg jelly. This is the nucleus and this is the acrosome. So upon meeting the egg jelly, there are some fucose containing polysaccharides which are initiating the acrosomal reaction in the sperm. Okay, the egg jelly contains some fucose containing polysaccharides. So when these will be releasing the contents over here, right? So after that, this will be sperm will be undergoing acrosomal process. So acrosomal process, it will elongate like this and they will come and bind with the next membrane, which is the vitelline envelope. Okay, so this is also acrosomal process, the extension of the acrosomal process. Now this binding receptors, they will interact with the sperm vitelline membrane, this purple color what you are seeing. Okay, then only the nucleus can enter inside the sperm. So afterwards, the cortical granule exocytosis will take place and the sperm probe nuclei, it will enter the egg cytoplasm. Right? Question number 4. So again, something about fertilization. So it's regarding chemoattraction, it's regarding exocytosis, capacitation also, and even the polyspermy block also. Okay, let's see Gokul. Okay, we'll see Anupam. So whether first statement is correct or not, everyone can tell here, isn't it? And if you figure out whether first statement is correct or not, anyways you will get your answer. Right? Confused between 4 and 3. Okay, so just figure out whether first statement is correct or not. Chemoattraction of the sperm to the egg is mediated by sperm activating peptides like bindin. So just now I mentioned what is bindin, isn't it? Yes, Dipshika, that's right, correct. Anupam, Gokul also, your answer is right. So it's a physiogenic protein. Now, why will bindin activate chemoattraction? No, chemoattraction is done by what? Remember chemoattractants. So what are that? Resact, Spiract, these are chemoattractants, isn't it? So this is how you should answer smartly. Okay, that's right. Uh, Correct, Anupam. And second of all is that means most important thing is bindin is present in sperm. But here it is written that it is mediated by sperm activating peptide like bindin. Okay, so it is present in sperm only. How can bindin present in sperm activate sperm? That's not possible, isn't it? So this is a very weird sentence. It is wrong. And if A is wrong means only this option doesn't have A. Rest all of them has A. So you have to say all the correct statement. So this cannot, any of these three cannot be there. So let's see the other statements. Exocytosis of the sperm acrosomal vesicle and release of enzymes will be taking place. That is correct because from the acrosome it is a modified Golgi complex, and from there many enzymes will be released. Okay, exocytosis and the release of enzymes will be taking place. The capacitated sperm undergoes acrosome reaction. Okay, so yes, because the sperm undergoes maturation only when it is inside the female reproductive tract. That is in mammals. So there they will undergo capacitation only after which their outer membrane becomes a little unstable. And that is how the acrosome will be able to drain its contents. Only the capacitated sperm can undergo acrosome reaction. This is correct. Okay, so the acrosome protein mediating, okay, no, sorry, so sorry. The capacitated sperm, okay, I think there is something missing over here because it is only B and E. Capacitated sperm undergoes acrosome reaction. So this is actually supposed to be correct statement. Okay, I will just have a check with this. Okay, I think something is in incomplete state sentence here because if only this much is there, then it's supposed to be correct. Okay, we'll just have a cross check in the next YouTube session class. Okay, about this third statement. The acrosomal protein mediating the critical species specific binding event is RISACT. Again, RISACT is not a acrosomal protein. RISACT is a chemoattractant. So what is said over here, acrosome protein, that is wrong. So D is also incorrect. Okay. And slow block polyspermy, it is accomplished by the cortical granule exocytosis. Yes, again this is correct because the fast block, it is by the membrane destabilization where it undergoes depolarization, and slow block, it is done only when the cortical granules release their contents. Okay, so E is also correct. In the next class I will just confirm about the C statement. Okay, so as I already told you, once the sperm binds with the egg cell membrane, so there is a kinase stimulation, and the kinase stimulation will be leading to, that is tyrosine kinase, it's a receptor. It leads to the phospholipase C activation, which will cleave the PIP2 into IP3 and DAG. IP3 leads to the calcium release, which is cortical granule exocytosis, and that is only what we are calling as slow block to polyspermy. Okay. Okay, next question. Let's move on to cleavage divisions. What kind of cleavage is shown in mammals? Okay, let's see. Rahul, Achal, Anupam. Yes. Okay, Deepshikha, fine. Okay, Achal has changed her answer. Okay, Aishwarya. Okay, Surinder has said one. Right. So don't be in a hurry to answer questions, okay? Especially the part B questions. Because many students they will do like that. Just because you saw holoblast doesn't mean you'll answer radial, isn't it? So yes, actually your second answer is right. The option is holoblastic rotation. Okay. So a lot of times I have told you that the mammals they have a very different kind of a cleavage division. First of all, they are extremely slow. They don't divide as fast as the other organisms. Second thing is they are synchronous. Their synchronicity is not present. So asynchronous. Okay, that means they don't divide together. These are the two very different style of mammalian division, and the third style is that they have rotational division, because mostly the cells will divide in same plane or like that, but here it will be different. So what happens is that the first cleavage division will be meridional only, okay, which gives rise to two cells. So this is one blastomere and this is another blastomere, okay. But what happens is the plane of division will rotate to form the blastomere. So after the two blastomeres are formed, the first cleavage, what was normal meridional division, but in second cleavage, one of the two blastomeres divide meridionally. So one of this is dividing meridionally, and the other one it will divide equatorially. So as you can see here, this daughter cell has produced two cells, one at the top and at the bottom. Okay, and in fact, this one happens little later. This one divides faster. Okay, and here because of the meridional division, they have produced one cell at the right and one cell at the left. Okay. Moving on to next question. The pattern of embryonic cleavage specific to a species is determined by two parameters. So the amount and distribution of yolk protein within the cytoplasm, and also what are the factors in the egg cytoplasm that influence the angle of the mitotic spindle. That means what plane the cell division will happen in, right? And also the timing of its formation as well. So which of the following statement is true? Okay. Saujanya says 1. So here any one statement will be correct, okay? Okay, Surinder says 2. What about others? Why am I not getting answers? This is such an easy question. We have discussed in class so many times. Anyone else wants to answer or should I explain? Okay, fine, Anupam. So there are some types of eggs we have discussed in the class. Yes, Rahul, that's right. So types of egg classification based on amount, you have alecithal. Now alecithal means there is no yolk. Microlecithal means very little egg is there, yolk is there, okay? Mesolecithal, moderate amount of yolk is there. And macro or megalecithal means lot of amount of yolk is there. Okay. So alecithal, no yolk. Microlecithal, negligible amount. Mesolecithal, moderate amount. And megalecithal, large amount of yolk is there. Right? So here are your examples: eutherian mammals, placental mammals. Then negligible will be tunicates, marsupials, and microlecithal. Mesolecithal is amphibians, major category. Other type is dipnoi and even lungfishes. So they are different from bony fishes. Okay, lungfishes have mesolecithal, whereas bony fishes they have macrolecithal. Okay. Apart from that, even reptilia, birds, Prototheria, Monotremata, these all have the polylecithal or megalecithal eggs. Okay, this is based on the type of egg. Now, how does type of egg control cleavage division plane? So we have something called as holoblastic cleavage, whereas in the complete egg is divided. If you see this diagram, this entire egg is divided. Any part of the egg is not uncleaved. Okay, and then further divisions happen. But in meroblastic cleavage, what happens is that you will see only a part of the egg is dividing. See, the rest part of it stays as it is. It does not divide. Okay, only at one part of the egg you can see the division. So this is what is an incomplete division of the egg. Now, why does this happen? It is because of the presence of yolk. So these parts are yolk rich. That's why the egg cannot cut through that portion. Now, while holoblastic is complete division of egg, cleavage furrow extend through the entire egg. Meroblastic is incomplete division of egg. Only a portion of cytoplasm is cleaved. So in holoblastic cleavage, furrow squeezes around the periphery like belt tightening, pinching the cells in two. And in meroblastic cleavage, the furrow starts at animal pole. It progresses into the egg like a knife and it stops when it encounters the yolk. Okay. So in what kind of organisms holoblastic cleavage will take place? Obviously, in the ones where yolk content is less. So that's why it will be taking place in alecithal, microlecithal, and even in mesolecithal. Only in the macrolecithal egg, that is mega or polylecithal, well, yolk content is more. Only there the egg cannot divide completely. So then only there will be a meroblastic cleavage. The word blastic comes because after cleavage division you produce a blastula. And mero means it's an incomplete cleavage dividing blastula. Okay, only a part of that egg will divide. Got it? So aim you have to remember as shortcut. Okay, alecithal, isolecithal, and mesolecithal. Whereas here you have two different type of egg. So under that you have one telolecithal egg you have, as what you get in these birds and reptiles and fishes. And another one you have centrolecithal. It is not mentioned here. So centrolecithal, it is based on the position of the yolk. So that happens in insects. Okay. So centrolecithal and telolecithal eggs you can remember as TC. So they undergo meroblastic cleavage, whereas AIM undergoes holoblastic cleavage. Now you must remember the organisms for that. Okay, like what undergoes what things. That I'll show you in the next question. Fine. So here I have given takes place in alecithal, isolecithal, and mesolecithal, and this takes place in telolecithal and centrolecithal. Okay, so as you can see here, species having. So now, can you answer everyone? What will be the answer now that I have explained to you? Yes, Deepshika, that's right. Okay, so only the isolecithal egg they will follow a holoblastic cleavage because only they have little amount of yolk. Yes, Aishwarya, that's right. Telolecithal has heavy content of yolk, which is called as concentrated at one end of the egg. So how can that go complete cleavage? It cannot undergo complete cleavage. Hollow word comes from whole. Okay, it cannot happen. Central acetal also has yolk at the center of the egg. It covers quite an area, so that also cannot undergo holoblastic cleavage. Isolacetal egg follow a meroblastic cleavage. Okay, so isolacetal eggs, they have very little yolk, which is distributed equally. So why will they undergo incomplete? No, they undergo complete division. So again, I want to remind you about the Watson Scholarship because tomorrow is the last date to apply. Okay, so you know what all perks you're getting in that, and you should not delay if you want to be a part of it. Yes, match the following cleavage patterns with the species in which they occur. So now that you know what are the different type of cleavage divisions, major category, that is holoblastic and meroblastic. Next thing is about the types of representative organisms. Yes, come on. Just now I said about the insect, right? Birds and insect, at least you know what type of egg they have. So you can predict what type of cleavage they can have. So one, you have meroblastic discoidal cleavage pattern. That means the cleavage division will be like a disc. Okay. And superficial, meroblastic superficial, the cleavage division will appear like only on the periphery. Okay. Surinder Aishwarya Rahul, yes, you have answered. Good. Let's see. Right. So definitely we now know that out of all this, just now we discussed the organisms, what all they are having, what type of eggs. So birds and insect, these two have the birds are having telolacetal egg and insect is having centrolacetal. So definitely for these two you have meroblastic. Even if you don't know which is what, okay, but definitely you know for TC it is meroblastic. Just now in the previous slide I showed you how to remember AM and TC. Yes, Achal Anupam, yes, all of you are right. Surinder, Aishwarya Rahul, Achal Anupam, all of you are right. So C and D will be 1 and 2. So you can see only in the first option. Okay, I think I did that by mistake. Yeah. So only in C and D you will have the option as 1 and 2. Okay. And frog, we know what kind of a egg you have. You have moderate amount, mesolecithal. Okay. So mesolecithal eggs, they will be undergoing holoblastic only, and even flatworm will be holoblastic. So because frog has moderate amount of yolk, okay, so little bit yolk is there at the bottom. So the top portion of the embryo will divide fast, and the bottom of portion of the embryo, it will be slow. So as a result, the cleavage division will be, we call it like it is slightly displaced towards the top. Okay, it will happen at the bottom but slowly. So that's why frog will be option 3. Okay, frog will be option 3, and flatworm is holoblastic spiral. Regarding the birds, so birds have discoidal. Why? Because if you saw the previous diagram, the cleavage division happens only at one end. Okay, only at one end at the top, it will appear like a disc. That's why it's called as discoidal cleavage. Whereas insects, they will undergo meroblastic superficial, which means that if you have the central part full of yolk, in the central part division doesn't happen. Rather, the division will be taking place in the periphery superficial. Okay, but that too it will take much later. After the 14 cell division in the insects, only after that it will be taking place. So these are the different holoblastic cleavage divisions. As I mentioned, this is for amphibians. So they were asking about frog, right? So it's a displaced radial cleavage, whereas in the top cells, they are growing much faster. So it's a displaced one, right? And in isoleucithal, yes, you have to remember all these representative examples. So radial cleavage is where it will cut either from the top portion like meridionally, or it will cut equatorially. So only two cleavage planes are there. Okay, only two cleavage plane. Either it will be top or it will be bottom, means in the center. Okay, from top to bottom or in the center. Not center means equatorially, like this, which is perpendicular to the animal vegetal axis. So this is the animal pole, this is the vegetal pole. Okay, so if it is parallel to that axis, it will be called as meridional. If it is perpendicular to that, it will be called as equatorial. This happens in echinoderms and amphioxus. Whereas you have the spiral cleavage, examples are annelid, mollusk, and flatworm. Here, the one division, after that, the second division will happen at right angle. Okay, so first division it will take place. The second division, just 90-degree angle, the second division will take place. So like this, they will keep on changing the cleavage plane right angle at right angles, and that will lead to a spiral type of division. Bilateral cleavage we observe in tunicates. So here, after the division, you will observe equal right and left halves. In the embryo, you will have after the cleavage division equal right and left half. Rotational cleavage I already explained to you. The cleavage plane rotates. Okay, so first is meridional. In the second one, it will rotate, and that's why one cell will be meridional, while other cell will be equatorial. Okay, and this will lead to a structure like this. Then you have meroblastic cleavage divisions. So what we saw is that central acetal, where because of the heavy amount of yolk in insects, the cleavage division doesn't happen properly, as in only the nucleus divides. There is no cell division. Only cytokinesis, karyokinesis takes place. There is no cytokinesis. So that leads to the formation of something called as a syncytium. Okay, so all this different nucleus, they will be they will be dividing, and then they will be moving towards the periphery. After moving towards the periphery, they will be surrounded by cell membrane. So that is what is central acetal. Okay. Whereas discoidal cleavage, fish, reptiles, birds are there. So as I said, the division happens as if it is in the form of a disc. You will observe at the disc. There is cephalopod mollusk which also undergo this type of division, that is discoidal, but then they also separate the cleavage area into equal right and left half. So that happens in bilateral cleavage. Then below given are fate map of two organisms and the pattern by which they undergo cleavage. So which of the following combination will be correct? So here I want to just show you the diagram. Now see here for animal, this is for one type of animal, okay, and they have undergone this discoidal cleavage. Then you have another type of organism. So this has undergone uh, this type. So it is just like a simple what you get, the blastula, what you get. Okay, here you have a type of cleavage where is only at the periphery you are getting. The cleavage division is only at the periphery. Okay, whereas here you are getting the radial one. As you can see here, getting the radial one. So which out of this will be correctly positioned, right? So what is actually a fate map? A fate map is a graphical representation detailing the fate of each part of an early embryo in the adult animal. Okay, so we will be doing the fate map early in the embryonic stage. So this is an early embryonic stage. All these colors actually we have put artificially. Okay, biologists they will put artificially just to color it and show it like a diagram that which part is fated to become which part of the adult organism. Okay, now we know that several germ layers are formed, that is outer germ layer ectoderm, inner germ layer endoderm, and in middle you have the mesoderm. Okay, so this outer layer, it is in blue color. This is happening in case of like all the organisms only. So this ectoderm is given by blue color. The ectoderm, wherever this blue color is formed, like in chick, this complete outer part, it becomes the ectoderm. Got it? In zebrafish, only a part of the top part becomes the ectoderm, like that. Yellow part, it becomes the endoderm. Okay, from which we get the gut uh, things, like the parts of the the parts of the digestive system. Okay, so the gut tube and all will be coming from endoderm, and the red color portions, they are called as mesoderm. So mesoderm means some middle layer will be there. Okay, now if you can see here for chick, it is like this. So this is what the diagram that you are seeing over here is for mouse. Okay, so these two are for mouse. You can see here also, this is for mouse and this is also for mouse. Now mouse means mammals, right? So mammals will undergo what type of cleavage division they are showing over here, right? So what type, which one is correct over here? Mammals go for rotational. So B diagram is correct or D diagram is correct? Mammals go for rotational or mammals go for radial? Which diagram is correct? That's right, Rahul. Okay. So mammals do undergo the rotational. That means B is matched correctly. Fine. Yeah. So B is matched correctly. Very good. Correct. Okay. D is actually not correct. D is not correct. They don't go for radial. Fine. And then the other diagram, as you can see, it is for frog. So you can see it is similar to that of frogs. See, this is for frogs. Now frogs, they will undergo what type of division? So you have diagram A and you have diagram C. So frogs undergo what type of division? They go for discoidal or they go for superficial? Frogs go for discoidal. No, no, discoidal is for birds. See, even after explaining why you are making mistakes, everyone, how many times should I explain this part? We have discussed this in class also. We have discussed now also. Still you are making mistake. Discoidal is birds, no? And you are telling frog. So here I told you, no, amphibian. What is frog? It's an amphibian, right? So amphibian undergo what? It undergoes displaced radial cleavage. How hard is this to remember? Got it? So none of this is right. None of this is right. Frogs don't undergo discoidal also. Frogs don't undergo superficial also. In fact, they undergo displaced radial holoblastic cleavage. Okay? Displaced radial holoblastic. You have to tell whether it's holoblastic or meroblastic. Under that also there are subcategory. So subcategory of that is radial, and especially in amphibians it is displaced radial. Okay, so again by mistake the wrong option has been marked. So I will just take it over here. So answer is only B. Only B has been correctly marked. Okay, that is the mouse, it undergoes rotational holoblastic cleavage. None of this is correct. Everyone got the question? Everyone got it or not? Actually, this part was a CSIR question till here. They have just asked like which fate map is for what type of organism. Okay, that's all they have asked over here. This part I have just added just to check whether you are able to get or not. Yes, students, please confirm. Did you get this or not? Is this the The type of cleavage divisions are clear or not? Or do you want to see the chart once again? Okay, fine. So if it is clear, then I want everyone to answer properly this one. I want all the answers to be correct. So Saujanya, you have to mark either 1, 2, 3, 4, any of this. Or do you want me to explain superficial once again? Is that what you're asking? You have to mark any one of this option. So see, these are two different questions, okay? So that previous match the following was asked in 1 year, and this has been asked in the second year, in another year. So as you can see, I have been discussing so many questions with you. I mean, we have just two topics, that is fertilization and cleavage, and from cleavage itself you can see from fertilization also so many questions have come, which means that it is an important question. Okay, so do not skip this part. Okay, so I'll just discuss the answer with you. Anyone else wants to try? So I don't see how it is difficult now. Why I have got answer only from Achal and Deepshika? Where are other answers? I'll give you 2 more minutes. Everyone has to answer. At least try. What is this? Why are you not concentrating on the explanation? See, you are spending time to attend these YouTube live sessions, but if you don't revise your classes and if you don't pay attention to the explanation also, then you will not be able to solve the further questions, no? Yes, Tejashwini, Rahul. Okay, fine. So, yes, here the correct answer is option 4. Very good, all of you. Okay. So we know that we have the amphibians. Here again, we know that amphibians, they have the mesolecithal type of egg. We know they have. Okay. So they will undergo radial cleavage. Okay. So it's not meroblastic. You should see what type of egg they have given. So this is the type of egg. The first word is the egg. The second word is the type of division. Here they haven't mentioned holoblastic or meroblastic. They have only mentioned whether it is the subcategory they have mentioned. Okay, so don't think as meroblastic this one. This is mesolecithal, which means the type of yolk content what is there. Yes, Surendra, Aishwarya, Anupam, your answer is right. Then you have so B is 1. Okay, so B1 means this also can be, this also can be there. Centrolecithal, superficial. So centrolecithal means the yolk is at the center. There itself you have C4. So see, already you got your answer. Even if you don't know the other two, if you know only two of this match the following, you can still get your answer. Okay. Then A will be 3, isolecithal bilateral. It is in tunicates, and D will be 2. That is telolecithal discoidal. This will be in case of birds. Okay. One last reminder of the Watson Junoon Scholarship, where you get 1 year coaching for all the exams that you want, CSIR NET or GATE or QAT IIT and JAM. And GAT B, you will also get 12 months of access, full 1 year access to all the workshops that Biotechnica conducts, all the global and national workshops. You also get art programming internship with live project hands-on, 4 certification course of your choice from our library, direct access to our coaching experts on WhatsApp, and 1 VIP pass to our CRISPR conference that will be happening this year. The time is only till 15th August, last date tomorrow to apply. This is your toll-free number, and this is the number, office number, where you can contact. Okay. So I would like to show you one more question. It is not from CSIR, but you can still try answering that question. I think this was a very old question. It may be from CSIR also. But these questions keep on coming. Like this is for capacitation. Capacitation of mammalian sperm allow them to be activated within the uterus and facilitate fertilization. So following statements were made regarding events during capacitation. One is removal of cholesterol, removal of non-covalently bound glycoprotein, induced expression of fibronectin, and decreased permeability of calcium ion. Which out of this will be correct? So any one will be not correct. Okay, so Surinder Achal, both of you have mentioned option 3. All right. So here, correct option is A, B, and D. Okay. See what happens. The removal of capacitation factors happens during the. So I will just show you this diagram. Yeah. So we have the cholesterol molecules over here, and we have albumin proteins, which is present in the female reproductive tract. Okay. So as you can see, the albumin is taking away the cholesterol molecules. So definitely you have the plasma, you definitely have the removal of the cholesterol. Okay, so first one is correct, removal of cholesterol from sperm head. And even there will be some glycoproteins which are bound non-covalently uh, on the sperm membrane. Okay, so actually those are the in peripheral type of proteins, you can say. So even these will be removed along with cholesterol. Even these will be removed. Okay, and what happens is that, yeah, so because of this calcium, it will get inside. As you can see, the calcium ions, it is getting inside. Fine. And bicarbonate ion is also getting inside. So bicarbonate ion, and that's why you have increased plasmatic membrane permeability. Fine. Because of the removal, the membrane permeability, it increases. But since it is said it is decreasing here, so since it is saying it is decreasing here, yeah. So again, I think the position of this changed because of the font. Okay. Yeah. So because you have the increase of the plasma membrane permeability, because of this, the ions. Now, because of this ions, the adenylyl cyclase got activated, and calcium, because it came inside, it acts as a secondary messenger, and that also led to the activation of enzymes. Okay. Now, because bicarbonate ion came inside, the increased intrasperm pH will be there. All these physiological events lead to the capacitation of the sperm. Okay. So that's why this is not correct. Okay, because decreased permeability of calcium ion, no, it increases the permeability of the plasma membrane. Okay, and yes, even the expression of fibronectin, which is a part of the ICM matrix, so all these events, they will be taking place. Okay, yes, correct, Aishwarya. Okay, but here it's not about acrosomal reaction. It is about capacitation. Both are different processes. Please do not take them as two same process. Okay. So that's all for today. I'll again discuss some more cleavage division questions in the next class uh, regarding the model organisms. Okay. Sea urchin, sea elegans, amphibians in details. Okay. Thank you everyone for joining. Hope the session was beneficial to you. See you in another class. Bye.